This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 718, recorded on February 9, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is cold. It's 30 Fahrenheit, but it just feels extra cold. -1 Celsius and very gray and cloudy.
It's totally foggy. I can't see New Jersey across the river here today. So weird. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 20 degrees Fahrenheit, feels like 13, and that's -6 Celsius. -11, feels like -11. It's sunny and we had light fluffy snow, so it's all sparkly. It's just the best kind of sunny day.
And from Austin, Texas, Rich Condit.
Hi, everybody. The weather here stinks. In a relative sense. It's 58 and overcast. We're on the leading edge of a cold front that we're probably sending to you guys. And this is going to be significant from a Texas point of view. We're going to have 3 days with the nighttime temperatures in the low 20s. So,
Wow.
Cool.
Preceded by a few days of rain. So
We're getting ready for another snowstorm. We keep having them. I'm wondering if you're sending us that.
that's probably true. Yep.
Yeah, we had one. We have had two, right, Brianne? In
We've
two
had
weekends
two
in a row. Yeah.
Mm-hmm.
If you like what we do here at TWiV, consider supporting us. We'd love your support. Microbe.tv/contribute. Today we have 3 stories for you. We have 2 vaccine reports and a basic science report. So let's start with vaccines. The first is a preprint, single-dose administration and the influence of the timing of the booster dose on immunogenicity and efficacy of ChAdOx1, which is also called AZD1222 vaccine. They still call it COVID-19 for some reason, which we remarked on in one of the other papers by this group.
So many of these vaccines have several different names, none of which are memorable at all. Okay.
I think mRNA
like
vaccine
ChAdOx.
works for me. ChAdOx I like. I like ChAdOx, yeah.
Yeah.
So
It's
this
also
is the
known as the AstraZeneca.
AstraZeneca, yeah.
Of course, ChAdOx is the vector, right? So they can put different things in it. You got to give it. So they call it AZD1222, which is not memorable. But I tell you, mRNA 1273, now I know how many amino acids are in spike all the time, right? Which I could never remember. Anyway, this is from a big group, which includes the Oxford COVID vaccine trial group. Sarah Gilbert and Adrian Hill are all the way at the end here. Andrew Pollard and Marin Voisey is the first author, but there are lots of asterisks. Let's see, 1, 2,
There's
3, 4, 5 first authors. Voisey, Clemens, Mahdi, Wex, and Foligati.
I guess that there's about 75 authors.
Some very interesting things in here, and that's why we're going to talk about this. So remember that this is a chimpanzee adenovirus vectored vaccine that is carrying the gene for the spike protein. And we already heard about phase 1 and phase 2 results. We've talked about those before. We've heard of interim efficacy. So here, efficacy of 2 doses was demonstrated at the interim analysis of 131 cases from data in Brazil and the UK. It was 70%. So the vaccine was authorized for emergency use in the UK on 30th December. This is very interesting. The Oxford studies were initially planned as a single dose, but were amended to incorporate a second dose after review of phase 1 immunogenicity data, which showed a substantial increase in neutralizing antibody with a second dose of vaccine. So that's interesting because people often ask, why do you decide to use 2 doses? And there you go. They tell us right there.
Yeah,
My
I was
sense-
so, I
Go
was
ahead,
so
Brianna.
excited to see this paper because I know how many times I've heard the questions about, well, what if we only did one dose to spread out doses, or what if we did our second dose later? And people have, asked those questions and hypothesized about them. And it's nice to actually have some data to start to address that on, instead of trying to come up with supposition.
My sense is that all of these vaccines have some benefit, at least in terms of immunogenicity, that is, antibody, of a boost. And a lot of this, some of this depends on how you want to structure. A lot depends on how you want to structure the trial. If you structure the trial to do an immunization and a boost, then you're unlikely to get good information about just the single dose.
Yes.
And if you don't have the information about the single dose, you can't use it in that protocol. And we got a pandemic going on. All right. So you'd like to be able to give people one dose. It's a complex problem.
Right. But at the same time, if one dose isn't enough and you didn't do the 2-dose trial, then you just got a vaccine that, didn't work, that maybe would have worked if you had used 2 doses.
So in a way, when you make that decision, setting up your trial, it's kind of a gamble. Okay. 2 doses is probably going to give you the optimal efficacy. Okay. But if you think maybe you could get away with 1 dose, you might want to structure your trial that way. Okay. It's a gamble.
So, as you say, this is an emergency. Normally you would have time to do it multiple ways, right? So go ahead, Kathy. Go ahead.
I was just going to say, before we get too far, this is only a preprint.
Yep.
So we have to remember that it has not yet been peer reviewed.
So there are 2 aspects of this trial which are interesting. First, they say, after providing consent to participate in a single dose study, some participants chose not to receive the second dose, providing a self-selected cohort of single dose recipients. Nah, I'm not coming back. Okay. And then additionally, due to the time required to manufacture the second dose, there were delays in administration for a large number of participants in the 2-dose schedule. So they say these 2 situations provided an opportunity to explore the immunogenicity and efficacy of a single dose and the impact of an extended interval. And they have now data from an additional month of follow-up. So that's kind of 2 accidental situations contributed to them being able to do this. It's interesting. I'm glad they told us, right?
Yeah. In addition to the other accidental situation, which was that there were some people that got the smaller dose, the lower dose.
Yes.
Which is going to turn out to be more complicated than just that, because that same group had a longer interval between the first and the second dose.
Yeah.
Right.
Yeah. All right. So the results: 332 cases of primary symptomatic COVID occurring more than 14 days after the second dose, the booster dose. So in the SDSD, that's standard dose, right?
Yep.
74 cases occurred in the vaccine group and 197 in the control. That gives you a vaccine efficacy of 63%. In the low-dose standard dose cohort, we have a vaccine efficacy of 80.7%. So overall, 66.7. I don't know if you can calculate an overall.
I don't know. I don't
think you
that
can.
doesn't make any sense to me.
Yeah, it's 2 different doses, so you got to keep them separate. 80% is obviously better than 63. And I still haven't heard a reason why this would be, right? We have speculations, but nobody really knows yet.
Well, as Rich said, they're going to point to something in this paper.
Yeah.
Okay, 130 cases of asymptomatic infection occurring more than 14 days after the booster. In the standard dose cohort, no evidence of protection. Vaccine efficacy of 2%. In the LDSD, there were 47 cases and the efficacy was 49.3%. So these are asymptomatic infections.
So 2% in the standard dose and 49% in the low dose, both followed by a standard dose for the second dose.
Right. Then overall reduction in any PCR positivity. was 54%. And they write, indicating the potential for a reduction of transmission with 2 standard doses. And the logic there will come into in a bit.
So I read this 2 ways. As sort of a good news, bad news situation in a way, or just news and news. Okay. On the one hand, there is a reduction of PCR positive infections. Okay.
Right. Yeah.
Apparently. Okay. On the other hand, there's a lot of apparently asymptomatic infection happening in vaccinated individuals. So this speaks to this notion of whether or not there's going to be, well, it's a clue in the question of whether or not there's going to be transmission in a vaccinated population. It is, I think the bottom line here is that it is definitely not sterilizing immunity in the majority of the population. It's a significant fraction of the vaccinated people are experiencing virus replication after immunization.
So can we clarify one thing here? So they talk about the difference between asymptomatic infection and PCR positivity.
Yeah.
How are they defining those two things as being different?
So they say that this is in the discussion. Actually, let's bump ahead and get their words. Okay. This is their logic. Okay, here. While transmission studies were not included in this analysis, swabs were obtained from volunteers every week in the UK, regardless of symptoms, to allow assessment of the overall impact of vaccine on risk of infection, and thus a surrogate for potential onward transmission. If there was no impact of a vaccine on asymptomatic infection, it would be expected that an efficacious vaccine would simply convert severe cases to mild and mild cases to symptomatic, with overall PCR positivity unchanged. A measure of overall PCR positivity is appropriate to assess whether there is a reduction in the burden of infection. That's their logic for why they think that is giving us some clues.
Right. No, I get that. I'm saying that if I go back to where I just was, I've scrolled around too much now. They are stratifying a group who has asymptomatic infection, or those who do or do not have asymptomatic infection, and those who are PCR positive, because they're making a measurement based on PCR positivity, and they're making a measurement of symptomatic infection. So there were 130 cases of asymptomatic infection, and then they also talk about the overall reduction in any PCR positive.
Yeah.
Yeah, I had the same difficulty, Brianne. What the difference is between those groups, or whether one group is included in the other. This is in table 1. They summarized this appropriately, and I can't differentiate between those. I don't understand that.
Well, their idea is that everything's going to be PCR positive, symptomatic or not, right? So if you see a decrease in total PCR positivity, they're saying it suggests we're reducing transmission of all those infections we're not picking up by symptoms.
Okay, so the asymptomatics are in PCR positive, but so are the symptomatics.
Yeah, that's right. They lumped them together for that analysis, yeah.
Okay. Okay.
Right. So it's a assumption. It's an assumption for sure. They say that, right? That's reflecting a decrease in transmission, but the study was not designed to do that. So they're trying to look at the data and get some clues. Okay.
Yep. That's another.
Yeah, I get it. I get it now.
Yeah, I get it now too. Speaking of the study not being designed to do that, there are several of these subcategories that have very few cases in them.
Yeah. That's
okay.
Right.
So it's I'm not bashing them, because I'm happy to see them split these out and report what's going on with appropriate qualifications as to what you can make for conclusions. But some of these don't have a lot of cases in them.
All right, another bit of results. Protection against primary symptomatic COVID with a single standard dose vaccine was modeled against time since the first dose and showed no evidence of waning of protection in the first 3 months after vaccination. And a single standard dose provided protection against COVID in the first 90 days of 76%, but did not provide protection against asymptomatic infection in the same period. So that's interesting. So it will protect against disease, but not infection, apparently, right?
Yep.
That's a single dose, yeah.
That's a really important piece of data. We've been talking about this for months.
Yeah, absolutely.
People are asking, am I going to have to wear my mask once I'm vaccinated? And we say, well, we don't know, but there's a possibility that even if you're even if you're resistant to disease, you may be able to be infected and potentially transmit virus. This doesn't measure transmission, but it does measure virus positivity, apparently, or PCR positivity, if you want to get technical about it. But it indicates that potentially people could spread virus.
And that's true here with one dose.
Yeah.
With one dose.
And so, that could be a reason to advocate for 2 doses. But we need to see those data.
So remember, I said the overall reduction in PCR positivity was 54% with 2 doses. Now they say single dose, the overall reduction of PCR positivity is 67%. So better after a single dose, which they say suggests the potential for a substantial reduction in transmission, which is, to me, it's counterintuitive, but I guess asymptomatic infection can be separated from transmission because that's what they're doing. They're saying, all right, we don't have protection with a single dose against asymptomatic transmission, but we do have a reduction in overall PCR positivity by a single dose. So it's a little, for me, it's a little uncomfortable.
There are aspects of this manuscript that I found very unclear. And one of them that was frustrating early on was that it's a confidence interval. And instead of saying like 52% to 72%, they put a comma in between the two. And it's an interval. But the first time they did that,
that's right.
I actually thought they were referring to 2 different entities, low dose, standard dose, standard dose, standard dose, but that they had just left out the word respectively, and so
Maybe it'll get fixed in the when it's published.
I don't know.
Maybe not. These things are coming out so fast. We did a paper, a preprint like last week, and it was just published yesterday.
Wow.
All right. Now, the other thing here is modeling of the change in efficacy against COVID showed that efficacy was high after a 2-month interval and continued to increase with longer dose interval. So this is between first and second doses. They had some people who took longer than others, or because of the manufacturing, it took longer to get the second dose, right? Vaccine efficacy after 2 standard doses rose from 54.9% less than 6 weeks to 82.4% when spaced more than 12 weeks apart. And, in the discussion, they say this validates our government's approach, right?
At least in a pandemic where we haven't done totally all the studies that we might have done.
Yes, right.
Right. And of course, this is in that low dose, standard dose cohort.
Yes.
Not in standard dose.
That's right.
No, wait a minute. I'm looking at table one again here. Standard dose, less than 6 weeks, 55% efficacy, 54.9. Greater than 12 weeks, 82.4%.
Right.
There's an issue going on here in Austin where shipments of vaccine that is designated as second dose is delayed. Now, this is either Pfizer or Moderna, different vaccine, but I think the same principles are likely to apply. And, some people are understandably freaking out because they're not going to get their second dose on schedule, and they're wondering whether their whole vaccine thing is going to be compromised because of that. And this suggests, at least with this vaccine, no, that's not the case. You're actually almost better off if you wait a little longer.
Well, however, Rich, in Austin, you have to keep it weird, right?
Yeah.
Also, in the UK, they are doing the same thing with the Pfizer vaccine. They're delaying it to the 12 weeks. So we're going to have probably a bigger cohort than just Austin that will give us data about that. But I think if after 2 doses, the efficacy is 94 or 95%, can you get much better than that by
No.
extending out the time? Maybe not.
True.
And
Yeah.
do you lose participants by expecting them to come back in 3 months, whereas in 3 or 4 weeks, they're still around and, where they got their first dose and all that?
Yeah, I don't know that I would translate these data with the ad vaccine to an mRNA vaccine, because there's the additional issue with the ad-based vaccine of immune responses to the adenovirus.
Okay.
And so I would wonder whether the spacing issues might be different with the adenovirus vectored vaccines because of that additional complication.
All right.
Yeah, fair enough. Kathy,
Good
can
thought.
you remind us what would be the seropositivity against chimp ad26 in the general population?
Well, the one place where we have data for that I remember is in Ghana, where it's 9%, and they compared that to Europe, I guess, and that was 0%.
My recollection is that in the UK it's 0, and actually
Yeah.
Gambia, I think it's
Or Gambia, yeah, sorry.
6 or 9%, something like
So
that.
this is
Yeah.
chimp ad, not ad 26,
Chimp ad.
right? Chimp ad.
ad.
Okay.
Right.
Add 26, add 26, depending on where you look, it's somewhere between 10 and 90%.
Right.
Exactly.
Sorry, I mixed up the 2 vectors.
Well, and the chimp's original name was Y25. So 25, 26, but yeah.
All right, so this pattern of having better efficacy with a longer interval, they say efficacy against asymptomatic had a similar pattern, but we have too few cases and they have really big confidence intervals. So yeah, you have to take with a very large grain of salt. Let's see. I also looked at neutralizing antibodies, which are higher after a longer interval before the second dose. Plotting the spike IgG against vaccine efficacy for each dose interval showed a clear relationship between binding antibody and vaccine protection, as well as between neutralization antibody and vaccine efficacy, suggesting potential correlates of protection. There you go. So those are the data.
So I take away from this that 1 dose works pretty well,
Yeah.
quite well, that you can wait significantly longer than 3 or 4 weeks, up to at least 12 weeks before getting a second dose, and you don't lose anything. And that it's in a large fraction of individuals not sterilizing immunity. You can still get an asymptomatic infection. 3 really important points.
Yeah. I mean, they make the argument that when you have limited vaccine, maybe we should get some broader protection rather than half of them. And they say they've done modeling, which suggests even if the first dose wanes, if you delay the second dose to vaccinate more people, you get a better population protection. Okay? Models. They're models. So they decided to do that in the UK. Now here we're not, except when you have to, like in Austin, because the vaccine is late.
Well, we're not. We're not in trouble yet.
Yeah,
Okay? It's not on schedule. It may not, in the long run, be late or significantly late. A couple of days, maybe.
Okay. All right. So, so this low dose, standard dose regimen, which was suggested earlier, they say it's confirmed in the current analysis. We have more data, and we think it's partly driven by the longer dosing interval that was a feature of this group.
Right.
So that's the idea, that the LD had a longer dosing interval, and that's why it worked better. It's interesting.
So, but we don't. I mean, that's a hypothesis. I mean,
Yeah.
that fits.
Right.
Okay.
Mm-hmm.
It did have a longer dosing interval. A longer dosing interval might give you better efficacy. So that might explain it. But
Right.
there are other possibilities as well.
Right. You need to do the experiment.
If you look at the graph in figure 1, that's the overall data, or just the standard dose, and then where the low dose, standard dose points come, then maybe that efficacy is quite comparable at that time point for the standard dose that had that same longer interval.
Yeah.
It's kind of a visual way for me to see it.
They say that we prefer the SD. It's more straightforward to deliver a single dose.
Oh, yeah.
Yeah. But when we get to the next paper, they're giving two completely different viruses for the two doses. So how hard is it? I don't know.
I don't think it's, you go separately. It's a different, what's the difference? You get a card that says what you got when. I mean, I think it's okay. The last thing I wanted to point out is this idea of we may have a hint of a correlate of protection. So they say our data suggests that humoral immunity may play a role. In contrast, high protective efficacy early after a single dose suggests other immunological mechanisms are at play because there are lower levels of neutralizing antibody after a single dose. Yet you have higher protective efficacy. Hey, is it possible that there's something else besides antibody?
What?
Shocking.
No
Isn't it shocking?
way. There are these things called T cells I might have heard of.
Mm-hmm. Shocking. Other things too, right?
Other things too.
Yeah, other kinds of cells. So a lot to be learned. Nevertheless, as they say, where is that sentence where they justify the government further? Here we go. These analyses show that higher vaccine efficacy is obtained with a longer interval between the first and second dose, that a single dose is highly efficacious in the first 90 days, providing further support for current policy. I wonder if Boris Johnson reviewed this manuscript and liked it. No, I don't think so.
I learned that his nickname is Bojo, or maybe Bozo, but mostly Bojo.
Okay, then this paper we just talked about was actually queued up for Friday, but we never got to it because our guest talked for 1 hour and 45 minutes, which was fine. It was great. So now we have this paper I also had found last week, but this is the Russian vaccine Sputnik V, safety and efficacy of an recombinant AD26 and recombinant AD5. There's the AD26. Vector-based heterologous prime boost COVID-19 vaccine. An interim analysis of a randomized controlled phase 3 trial in Russia. So we talked about the phase 1-2 results of this vaccine earlier. This is a combined two different vectors, right? 26 and 5. You get one first and you get the second. And Kathy, so did you already tell us the seroprevalence of these before? I don't remember.
It depends where you're looking, but overall for AD5, it's probably around 86%. That includes Brazil, some countries in Africa, Thailand, Europe, and the US. Whereas for AD26 overall, it's more like 58% in those same countries. But for AD5, there's a much higher neutralizing titer overall in the population than there is in AD26 as well. So doesn't fit into those percentages.
This heterologous vector strategy is an attempt to get around the vector immunity problem, where theoretically, if you have a virus vectored vaccine and you immunize somebody, they make antibody not only to the antigen that you're expressing, but also to the vector, so that if you try and boost them with the same thing, antibody that you made to the vector may compromise the boost, by stepping on the vector. And I think that there's a lot of evidence from a lot of trials with AD5 that can be a problem. A problem not only with vaccine-induced immunity, but also with pre-existing immunity, because AD5 circulates in the population. AD26 is a different story. First of all, one of the reasons that Johnson & Johnson, for example, started out with AD26 was the presumption that it would have a fairly low seropositivity in the population. But then when they looked around a little more, they found, well, no, it's pretty variable. And in some places, it can be as high as 90%. But as luck would have it, either the natural immunity or the vector-induced immunity, for some reason, doesn't seem to significantly compromise reutilization of the same vector. Okay, that's why J&J ultimately has gone with a prime boost with the same vector. Okay, you could say that the heterologous vector administration is not, well, if you want to use AD5, then it pays to use another vector second time around. But apparently you can use AD26 twice with no particular problem.
So the one thing I will say is, in addition to that, is at least in the HIV vaccine development field, the idea of using heterologous prime boosts, where you use two different things, either say nucleic acid for prime and a vector for a boost, or two different vectors, not even necessarily two different ads, but two completely different viruses as vectors, has been a very positive seeming approach. That approach has gotten good data in general. And so the idea of a heterologous prime boost for a vaccine is one that has some support in the literature.
Right.
And part of the reason to try to stick with AD5 is that it is really good at being immunogenic. And so I think we talked about, Sinovac in China is maybe AD5. And somehow they're able to do that.
Kathy, this is in the very back of my memory from being in grad school, where people were talking about adenovirus vector-based vaccines. So you might know this more than I do, but is there also a difference in how well some of these adenovirus serotypes express a transgene?
Oh, yes. I think that's part of it, for sure. Why they're more immunogenic is because they're better at expressing the transgene. I may not have said it that way, but I think that is definitely the case.
Because I think that's the thing, is that AD5 expresses transgenes the best, but it also is particularly immunogenic. And so there's sort of a trade-off.
How is chimp at expression of genes, Kathy? Is it good? Not as good as 5?
I don't know. I haven't seen anything on that lately.
Do you know if there are other chimp ad serotypes besides the one they use?
There are. So chimp ad 7, chimp ad 6, and chimp ad 65, I thought I saw. And they talk about the seroprevalence for them too. Yes. Okay. Also, porcine, bovine, ovine, they were in one paper that I saw this morning. So a lot of other possibilities.
Okay. All right. So this trial, 18 years and up, 2 doses, 21 days apart, 10 to the 11th viral particles per dose in your muscle, intramuscular. Of course, there's placebo group as well. And then they tested people by PCR initially, and then if they had symptoms, they would confirm them with PCR, but no other PCR tests, so no possibility for looking at asymptomatic infection here.
In fact, they had quite strict inclusion and exclusion criteria. So they tested everybody by PCR before they started. And then a couple days later, evidently, is probably when they gave them their first dose, because they had to have time to get the result.
That's right.
And if you were PCR positive, you weren't even allowed to be in the trial.
Yeah. So 21,977 in this trial. And then 5,000, so split between placebo 5,400 and vaccines 16,500.
So about 3 to 1?
Yeah.
And I love how they refer to database lock.
Yes. Yes. It was very clear what they meant with that. It was good.
All right. So from 21 days after the first dose, which is the day of dose 2, 16 COVID cases were confirmed in the vaccine group and 62 in the placebo, which gives you vaccine efficacy of 91.6%. So that's pretty good. Now, you remember when we talked about the phase 1/2, we were critical because they only had like 70 participants total. So now they've done a proper phase 3, and it's looking pretty good.
So that's just that prime. This is just the first dose. Right?
That's right.
And that's the
Correct.
AD26. And that's giving them, after 3 weeks, 91.6% efficacy. Yeah, that's great.
And in fact, efficacy against moderate or severe in the same, at least 21 days after dose 1 is 100%. No cases seen. 97 cases have confirmed in the vaccine, and this is not reflected in this analysis because they occurred fewer than 21 days after dose 1. But nevertheless, that efficacy is 73%. Anytime after dose 1, 73%. And they say in the vaccine group, most cases occurred before dose 2. If you look at the curve, you can see that. So dose 1 is giving pretty good protection. 16 to 18 days after dose 1, after which early onset of protection led to the number of cases in the vaccine group increasing much more slowly than placebo. And they have that Kaplan-Meier curve, which we've now seen multiple times. Very impressive, right? Vaccine goes up a bit and then boom, it's flat after a certain period of time. They looked at adverse events, of course, flu-like illness, injection site reactions, headache, asthenia, the usual. They had 4 deaths, 3 in the vaccine group and 1 in the placebo group. But they say they're not vaccine related. In the vaccine group, 1 death was associated with fracture of the thoracic vertebra. And the other 2 were by COVID deaths. Patients died of COVID. Because, not everyone was protected, I suppose, right?
Right.
Yeah.
Right. These are the ones that got it within the first 4 days. So between when they got tested and didn't get kicked out of the trial, they became positive and started showing signs and positivity.
Yep. Yes. All right. That's basically it.
I really like this paper, especially after reading the previous preprint one. This one was really well put together. Their confidence intervals are separated by a hyphen. And I just thought it was just really well laid out. It was easy to understand. And I thought they included a lot of things that weren't in the other paper, but they had different kinds of things to be presenting. The one thing, when I was talking about their exclusion criteria, they excluded people if they had extensive tattoos on their upper arm. So, yeah. Now
You know
I can't
why
find
that
it again.
is?
Why is that?
Maybe it makes it harder to read an adverse effect.
I think so. I'm having a hard time finding it again, but I swear I saw it. Yeah, large tattoos at the injection site.
Interesting.
Yeah.
But they say here again, so some suggestion of efficacy after the first dose. They say the cases diverge after 16 to 18 days after the first dose. And they say, of course, we can't draw conclusions, but it looks pretty good. The other thing I wanted to point out, of course, they measured antibodies here, and they also measured T cells as well. There were a few non-responders in the vaccine group. 6 out of 342 people did not make a response, which I thought was an interesting number, right? And
Did not make a response based on antibody, right?
Antibody and T cells, I think, yeah. So 6 out of 342, and they say immunosenescence in the older people, maybe people with immunological disorders, maybe random. But that's good to know. That's kind of roughly the number, right? 6 out of 300.
It'd be interesting to, if they had looked at the AD26 titers before the study, to see if those people had particularly high pre-existing AD26 antibodies, and that was related.
So far, as of January 23rd, 2 million people have 2 million doses have been administered. And they're looking into a single dose regimen. They're doing a clinical trial. How many people in Russia? Does anyone know?
Nope.
Russian population.
Go look it up.
Russian, I can't type. SS. Russian population is 144 million. they have a ways to go. Well, so do we, I guess. So anyway, in the end, Sputnik V seems to be good, right?
Oh, I also looked up why they called it Sputnik V. And of course, the original satellite that started the space race was Sputnik, and then there was another one, Sputnik 2, and so forth. And after Sputnik 3, they didn't really officially have anything called Sputniks anymore, although the West sometimes would call whatever the USSR sent up, they would dub it, Sputnik 4 or 5 or whatever. But I think just to be safe, they skipped one and then called this vaccine Sputnik 5. And, it does, an adenovirus does kind of look like a Sputnik, so it makes sense.
If you're wondering what Sputnik means, attendant, companion, or traveling companion, or fellow traveler. So if you go on a trip with someone, they're your Sputnik, right? Yeah, I remember Sputnik in the 50s, 1957, orbited 3 weeks, then fell back into the atmosphere. All right. Now we have a really interesting basic science paper in PLOS Pathogens. The coronavirus proofreading exoribonuclease mediates extensive viral recombination. And this is out of the laboratory of Mark Dennison, and the first author is Jennifer Gribble. And I really find this exonuclease interesting. And
can I give the overview of why,
go ahead.
why
Yes,
we find
please.
this so exciting? So you may have heard that viruses mutate, and you may have heard that RNA viruses mutate more than DNA viruses. And you may have heard that in a kind of unusual twist, the coronavirus, which is an RNA virus, has a proofreading activity which reduces its rate of mutation. And that evidence was kind of suspected for a long time because the coronaviruses have a much larger genome than the other RNA viruses. So how can they do that? Because they should suffer more hits per molecule than a shorter RNA virus would. So that's why it was probably thought that there was something different. And so Mark Dennison's lab, I think, was one of the main ones originally that identified that there's this protein that can serve as a proofreading enzyme. And it's part of the RNA replication complex, but it's not the molecule itself that we call the RNA-dependent RNA polymerase. It's part of the complex. And so it has this exonuclease activity, and that can serve to have it be proofreading activity. And there are some other activities that have already been attributed to it, which I'm not sure if I can find. But the gist of it is that in this paper, they are going to attribute another activity to this exonuclease N molecule. And that's what's so exciting. So
I want to even go further for the listeners and define what an exonuclease is, or at least try. Okay. So when we make RNA or DNA, you have a template, and you use that template to literally as a template, the polymerase enzyme moves along one nucleotide at a time and elongates a chain, matching up in a complementary fashion nucleotides. And it does it with directionality. You can distinguish one end of a RNA or DNA molecule from another. We call them 5' and 3' based on the chemistry, and the polymerization reaction goes from the 5' to the 3' direction. An exonuclease, a nuclease is anything that chews up nucleic acids, and you can imagine that you could chew up a nucleic acid by attacking it in the middle or attacking it from the ends. An exonuclease specifically attacks a nucleic acid from the ends. And there are exonucleases that work from the 5' end. There are exonucleases that work from the 3' end. This sucker is a 3' to 5' exonuclease. So it works at the elongating terminus of the synthesized nucleic acid, and it can trim off nucleotides in the opposite direction of the polymerization. And the way I think about this is that as a proofreading enzyme, if the polymerase is going along and it puts in the wrong nucleotide, okay, it actually kind of knows that. okay, because the base pairing's kind of screwed up and it may pause. And the exonuclease looks at it and says, and trims that out from the 3' to the 5' direction, excises that poorly base paired, the wrong nucleotide, and then the polymerase can go forward again. Okay, so it backs up. And in a lot of systems, these exonucleases can chew off more than one nucleotide, and that has implications for stuff like recombination. Kathy?
So I was just going to say, it's like kind of like a backspace key on a typewriter, or the delete key in a word processor, where you can go back one and then put in the right thing.
Yeah, excellent.
So the order of which the coronaviruses are, remember, nidovirales, they have these, they're unique in the RNA world, virus world, to have this exonuclease. Other RNA viruses do not have it. And they can't fix the errors that the polymerase makes. But some of them do recombination, and we'll talk about that later on. So we know that coronaviruses undergo recombination. It's quite clear. There's evidence in the past of various coronaviruses being recombinant. SARS-CoV-2 is clearly a recombinant. And the process by which the virus produces, or the polymerase produces mRNAs is in itself a recombination process. And I want to show a picture of that here because it might help those who are not driving. Now, this is a diagram of the viral genome on the top left, the 30,000 base plus stranded RNA genome. And then on the right are the mRNAs, which are nested. There's longer ones and shorter. They're all, as one of my students said, matryoshka mRNAs.
Matryoshka, yeah.
Matryoshka. And they are produced by a process where the polymerase moves to another part of the genome. And on the lower left is the model for that. We have the plus-stranded genome, and the idea is the polymerase starts at the 3' end, begins to make a message, and there's signals between these various open reading frames, transcriptional regulatory signals. And the polymerase then, after going a certain distance at the 3' end, at the right-hand part of the genome, continues all the way at the other end. The RNA actually probably loops around in some way, and then you can see it's shown in this picture by a dotted line, but it finishes each mRNA, so here on the left, with sequences from the very left-hand part of the genome. And that is recombination, when the polymerase goes from one template to another. And then that minus RNA is copied to make the actual mRNA. So this is recombination, and the polymerase complex is really, really good at it, which is why... go ahead.
I was just going to say, for people who haven't looked at this as much, if you look at those mRNAs on the right, in theory, all of them start with that little orange piece that you see on the left.
That's right.
And that orange piece has been copied and is on each of those. And they're sort of paired with a different part of the 3' or downstream region of the genome. And I remember the first time I started to look at this being sort of blown away by that.
Now the other,
so it's as if recombination in this form is an obligatory part of the coronavirus life cycle.
Absolutely.
And this is not, this is at least somewhat unusual. I mean, like poliovirus doesn't do this. This is for viruses like this that make subgenomic RNAs.
Yeah, the nidovirales will do this. Polio does recombination, but not by this mechanism, yeah.
So I have a question, Vincent. Do you know, or anybody, Brianne or Kathy, do you know what determines whether you make a subgenomic messenger RNA or whether you make full-length antigenomic RNA? Is that a stochastic thing, or is there some gene product that suppresses the messenger RNA synthesis?
I would guess it's probably the nuclear protein that does the switch, like it does for influenza and VSV and so forth. But I don't know for sure. Yeah.
Okay.
All right. So then, recombination also generates what are called defective viral genomes, where the polymerase skips, instead of making a faithful copy of the genome, it makes a shorter copy, missing genes. And those can replicate as long as they're in a cell that has the virus that provides the replication complex, defective viral genomes. And we'll see those today.
And for old timers that might remember defective interfering particles, sometimes these actually can interfere in the case of coronavirus. So we used to work on DI particles in my doctoral work.
And so in the coronavirus DI particles, they're going to be predominantly missing chunks from the middle, right?
Yeah.
And they'll have end sequences that are important in the replication and missing chunks in the middle.
That's right.
Correct?
Right. That's right. Mm-hmm. Now, the key here to this paper is they write, prior to the advent of next generation sequencing, direct analysis of recombined coronavirus RNAs was not possible, and the determinants of recombination could not be identified. And that's what this paper is, a lot of RNA sequencing. Basically, you infect cells, you can harvest all the RNA in the cell, you can harvest the viruses that come out, and you sequence the hell out of it, and then you use a program called Virema, Virus Recombination Mapper. We have to somehow make a title out of that. Virema, which will detect recombination events, which is basically, if you think about how recombination works, a little bit of the genome here and a little bit there joined together. So the program is perfect to go through and tell me where the recombination is. And they can quantify recombination. They can see what parts of the genome it's occurring, what kind of RNAs are made. So the mRNAs, of course, are all made by recombination. So they're picked up because they have the body joined to that leader part, as Brienne said. So they do this with MERS coronavirus and SARS-CoV-2, and later on with mouse hepatitis virus.
Now, I want to get something clear in my own head. They're sequencing sort of total viral RNA from an infected cell.
That's right.
And they're coming up with
polyA selected.
Okay. Yeah. And so they're sequencing the subgenomic messages. They're sequencing DI particles. They're sequencing full-length viruses. And there's no implication one way or another as to whether any given sequence is an infectious virus. Okay. Among all these sequences, there's going to be some replication competent viruses, but there's going to be messenger RNAs and DI particles and all sorts of other trash. Mm-hmm. Okay. But all that, if you sift through that trash, you get an idea of what the replication is like.
Yeah. And that's important because there are other ways you might think about this where you might not get to see all that trash. You might only get to see the things that made productive viral particles and how to sort of add this extra selection step. And here you're not having that selection step. You just get to see everything that gets made, even the trash.
And I was thinking about the trash today because, viruses can be pretty sloppy. As long as you get a few virus particles that can get out and infect another cell, it doesn't really matter how much trash you leave behind. And, I was thinking about these nature shows that I watch with my grandchildren that time and again go on about how many gazillion fertilized eggs some creature makes and how about only 1 in a million or 1 in a billion of those actually makes it to adulthood. Same thing. There's a lot of apparent waste, though it's food for somebody else, maybe.
So they do a number of things with the sequence data. The first is they say, is there a difference between MERS and SARS-CoV-2? And there is when you quantify the amount of recombined molecules. MERS has a mean frequency of 37 junctions. That's 2 different sequences put together that wouldn't normally be. 37 junctions per 10,000 nucleotides. And SARS-CoV-2, 475 junctions for 10,000. And they say this was surprising, and we should study it some more. And maybe cell type matters, maybe it differs in animals. Maybe it differs in people too. That would be interesting to look at.
So I was a little confused when I went into this because I have a preconceived notion of what recombination is, which is where you take two different parental genomes and you put them together in a cell and you look among the progeny for combinations of part of one parental genome and part of another. This is their sort of definition of recombination is different in this case. And as I understood it, correct me if I'm wrong, they looked for basically the mapper looks for deletions.
Yeah, that's right. That's right.
Where there's some missing nucleotides, and that is indicative of the polymerization reaction having joined two sequences that are otherwise not contiguous in the input genome.
Yes. If you did this experiment with two different coronaviruses or two marked, then you could see recombination that gave rise to infectious viruses. But if a SARS-CoV-2 recombined with itself, you would not know because the program wouldn't pick it up. There's no deletion. That's absolutely right. But you could put a marker on one genome or a bunch of markers throughout the genome and co-infect it with the other without it, and then have the program modified to pick that up. And then you could do what you do. Yeah, you're talking about old time stuff. We had actually
I wasn't going to say it, but you're right. It's old time stuff.
Yep. All right. So they're interested in this. And what do they see? They see certain areas of the genome, and you can see in these figures, it's quite nice. You can see where they map the recombination frequency according to where you are in the genome. The ends recombine a lot. The intermediate parts recombine a lot. Within the 3' end, 5' end, they get deletions and so forth. So really, you can go at a very detailed level here by sequencing it. They also find defective viral genomes, as we talked about before, where you would have the two ends joined together with the sequences missing in the middle. And they find that the highest frequency recombination occurs at where the mRNAs would be made, the junction of the mRNA with that leader all the way at the other end of the genome, as you might guess. But it's not just there. So there's way more recombination than just the ones that give you the mRNAs. It's all over the genome.
But I'm left wondering whether part of this isn't just sloppiness in that whole process. And what is, given that recombination is an obligatory part of this, okay, if there's a certain amount of slop in that, you generate all this other stuff. Now, I'm guessing here.
Yeah, it seems like it is energetically wasteful, but if the virus can do so much of it and only needs some of it to work out, maybe that's okay.
Niles always told me that there was plenty of ATP. Don't worry about the
Yeah, I mean, the other alternative is that maybe some of the trash gets used for something else.
Could be.
Yeah, but ATP is expensive. It's not cheap. It takes a lot to make it. They look at, all right, where are these junctions occurring? And they find evidence for areas of sequence microhomology, so homology between the two sequences that are recombining, which suggests the mechanism of what's going on. So they do quite a bit here looking at the architecture. But then the meat of this paper, in my view, is when they use a virus where they've inactivated the exonuclease. Mm-hmm. And here, the protein is inactivated. So they say we have no proofreading deficient catalytic mutant, so we inactivate the gene. I guess they make a deletion. So they're not making any XON. And then they infect cells with that virus, which will reproduce. It just is a bit wimpy, I guess. And it makes a lot of mistakes because it can't fix them. It can't fix its errors. And it has very much decreased recombination frequency. This is in mouse hepatitis virus now, where they have the XON deletion. That's right. They don't have it for MERS or SARS-CoV-2. They do have it for mouse hepatitis virus. That's right. And so that you inactivate XON, you reduce recombination that we have seen so far. You don't eliminate it, which is interesting, but you reduce it, which begs the question, where does the other recombination come from? right? Maybe it happens, maybe the polymerase just does it at a certain level and the XON stimulates it, right, in some way. Maybe, as you said, Rich, by pausing, that encourages recombination somehow. But anyway, recombination occurs.
I'm trying to refresh my memory on exactly the nature of this XON mutant, whether it's a deletion or not.
I remember it being a deletion, yeah. Well, he says it's we don't have a catalytic mutant, so we use the MHV, and that's reference 49.
Yeah, and they call it an XON inactivation mutant.
That's Eric Schneider, actually, apparently did that. It's a Journal of Virology paper, which I think, Kathy, you were mentioning, from what year is this? Why can't I find the year on the first page? Huh.
You say it's reference 49?
Yeah.
That's
enzymatic is critical for replication, Eric Schneider. Okay, so what they do, using reverse genetics, they made knockout mutations, blah, blah, blah. Okay, it's a substitution, E191D.
Okay, so, right. That to me is this is getting into the weeds here, but that to me is important because if, I don't even know if they know whether XON is part of a replication complex, but if it is.
Yeah, it is.
It is?
It is.
Okay.
Yeah.
If you're missing the protein, that could have pretty dramatic effects on the whole complex. If you're just tweaking an amino acid, you're looking at an effect on the activity. And so what we're learning here is it is not a deletion, right? It's a point mutation.
That's right.
Right.
Okay, good.
And so what that means is that this protein, NSP14, has two jobs, or more than two jobs, because Kathy told us earlier that it had a lot of jobs, but we've added this new job of recombination to the functions that are known for NSP14. And are there other examples of viral proteins that can act as both exonucleases and proteins that are involved in recombination?
Well, this is not quite the same, but in pox viruses, it is the DNA polymerase that is responsible for recombination, and the proofreading function is built into the DNA polymerase. And if you knock out the proofreading function specifically, that impacts on recombination. So we're talking about just a single enzyme there, but an activity that within that enzyme that influences both proofreading and recombination.
So kind of convergent evolution here in a way. It's like Vincent said that coronaviruses want to be DNA viruses.
The thing is, now I'd have to draw this out again, but as I said, my recollection is that the exonuclease activity can promote strand invasion for a homologous recombination event by exposing a single strand. And that's what I imagine going on in the pox viruses. This is different.
Yeah.
Well, maybe it's not.
Well, you have a double strand.
I don't know, because you do have a double strand, and I don't know exactly what the mechanism is here for what we're calling recombination.
Yeah, I'm not sure it's known.
Maybe it's similar. Well, anyway, in their discussion, they indicate that mechanistically they don't know what's going on.
No,
right?
They don't. No, I mean, all the recombination products are decreased. There doesn't seem to be any bias for any of the, defective genomes, subgenomic mRNAs, all the others. They're all decreased. They do see that there's a slightly altered junction site selection in the mutant, but basically all of the kinds of recombinant molecules are decreased. So basically, you alter the recombination landscape without XON activity, but it's still there. It's still there, and I think that's quite interesting. So this is the first demonstration that this XO is involved in recombination, and figuring out what it's doing would be really interesting. to look at. And they also say something at the very end here. They say, if you make these exo-N mutants, they're genetically, they're attenuated in animal models, the SARS-CoV is. And they say maybe it has less potential for recombination-mediated or even mutation-mediated escape from whatever it is, antibodies or such. So if you made a vaccine strain, maybe you make it in this background and it has less genetic variability. It's kind of an interesting idea. I know that people have thought of using this backbone for years for vaccines, but obviously we're not using it now.
I want to go back to this notion. I had a thought about this notion that making all the trash is wasteful, okay? Because there's an advantage to doing a lot of recombination because you make new variants. So that could assist in evolution. You have opportunities to recombine vat viruses and infect the humans, for example. What might seem wasteful, in fact, is you're just doing a whole bunch of experiments, okay, and selecting out of those the experiments that worked.
Wasteful is a human thing, right? We view it as wasteful, but I think we shouldn't do that. What viruses are executing may be a very different thing. So I wanted to show a picture of the poliovirus polymerase. This is the three-dimensional structure of the poliovirus RNA-dependent RNA polymerase. And because there are two single amino acid changes in this enzyme that affect fidelity and recombination separately. So for these enzymes, the recombination is controlled by the polymerase as opposed to the coronas that we've just discussed, where the recombination and the fidelity, the recombination is controlled by exo-N. And of course, exo-N can fix errors. But the fidelity of the actual polymerase doesn't change. It still makes the same number of mistakes. They're just fixed. So here, the polio polymerase, the active site is shown in yellow. So the RNA comes in from the right, and it goes to the active site, and then it goes up towards the right. And if you notice in the upper right part of the molecule, there's an orange set of atoms highlighted, and that's a single amino acid change that decreases recombination by this enzyme. And so a certain part of the enzyme is involved in controlling recombination. Then on the other side of the molecule, on the left side, there's another orange set of atoms highlighted. And if you change that, that affects the fidelity of the enzyme. If you make one G64S is the amino acid change, it will improve the fidelity of the enzyme, makes less mistakes. But such an enzyme in a virus is wimpy. It's not fit at all. It can't compete against a wild type enzyme. So making mistakes for a polymerase is good up to a certain point, of course. All right, so both of these changes, or amino acid changes, are mentioned in the paper they probably impact the one that affects fidelity. As bases go into the active site, when you have the right, they're all going in and out really quickly, all four, because they come in through a tunnel on the back of the enzyme. And then when the right one is in there, which happens randomly, it matches the template, then the enzyme undergoes a conformational change. And this one amino acid change is thought to make it harder to do that. So it has to match, because sometimes the wrong one gets in and it still works. But this change is thought to make it more stringent. And then the other one that affects recombination is thought to affect the speed of the enzyme. It kind of slows it down. And so, I don't know, it's walking on the railroad tracks and it's careful not to fall off, whereas the wild type enzyme goes really fast and it falls off a lot and goes onto the other strand, I guess. Anyway, so I just wanted to point those out in the polio polymerase, where the enzyme controls both fidelity and recombination. Okay, and so you may say, what does this have to do with the pandemic? Well, it's...
recombination gave us the pandemic virus. How about that?
This is what regulates it. And in the next one, maybe this information is going to be useful. All right.
One fun fact I wanted to point out about this paper is that some of the work was supported by the Dolly Parton COVID-19 Research Fund.
That's so cool.
So
Yeah.
you probably heard about that in the news, but yeah, that's pretty cool that they were able to cite that.
That's very nice. Let's do a few email here. The first one is from Dirk, and Dirk was on the Tempus paper that we talked about last week. Thanks for highlighting the Tempus 2 versus endocytosis story. I want to add that SARS-CoV-2 also adapts upon passaging in Viro E6 cells with amino acid deletions near the S1, S2 boundary, 5 to 10 amino acid deletion. This results in virus that replicates very well in Viro E6 cells, high titers, good CPE, but hardly on Tempus 2 positive cells like KLU3. The virus seems to adapt to the endocytosis pathway, loses the ability to enter using Tempus 2. These types of deletions do not seem to occur in humans or when passaging the virus on KLU3 or primary lung cells. Yes, I remember that. It was published last year, and that's, of course, because Viro 6 don't have Tempus, so it loses the site. Depends on the cathepsin inside the endosome instead. We screened compound libraries for antivirals on Viro E6 with the viruses containing these deletions, and like many other groups, found cathepsin inhibitors and other endocytosis-related stuff, PIK-FYVE inhibitors. So it is clearly something to keep in mind when evaluating therapeutics. Probably the story is even more complex, as the fetal bovine serum used in Viro E6 cell culture inhibits all kinds of extracellular proteolytic activities, pushing the virus even more towards endocytosis. We hope to optimize our systems towards virus stocks without deletions, serum-free medium, and Tempus 2 positive cells. This is probably best for screening, but don't know yet if it's feasible. Yeah, I mean, that could be a problem screening for inhibitors, right? You're going to get something that's irrelevant. Note, a virus with these deletions might be strongly attenuated and could serve as a live attenuated vaccine candidate. There are other vaccines produced on Viro cells, so this could be feasible. Cool. That's very interesting. And Dirk provides a few references about this. So thank you. It's always nice when the authors are listening. Kathy, can you take the next one, please?
Andy writes, and he sends a link about influenza update. Dear Vincent and colleagues, I listened on TWiV 709 to your discussion about flu rates in the US based on CDC data. I'm attaching the WHO influenza update. I think the first paragraph is particularly relevant to the graph that Kathy commented on. The data there were looking at influenza based on a % of total visits and is therefore subject to a variety of potential biases. It would be much more useful to look at absolute numbers of cases or at the very least of total visits. WHO makes the point that health-seeking behavior has changed. In the UK, you would be virtually excluded from a primary care consultation if you had flu-like symptoms because you would, by definition, have COVID-19 symptomatology and would be asked not to visit your primary care provider. I have, however, also attached UK weekly data on influenza and COVID monitoring, which does suggest that the inference from the CDC data may well be correct. Data looking at hospital admission rates and intensive care admissions due to influenza in the UK are below threshold for expected levels. Graphs from pages 46 to 50. The data on sentinel monitoring for respiratory viruses shows COVID and rhinovirus are around in the population, while there are a few cases of flu A and B, and virtually no respiratory syncytial virus or adenovirus. As a pediatrician for 35 years, I cannot recall a year when respiratory syncytial virus rates have been so low. Page 75 includes international reporting data on influenza. Flu lurks in the shadows. Keep up your good work. I hope you find these weekly data useful in monitoring the situation. It also includes some useful graphs on excess death rates in the UK. Best wishes, Andy.
So go ahead.
I was just going to say, this is really interesting. There is currently a COVID-19 a year later Keystone meeting going on virtually yesterday and today that I've been attending. And yesterday I saw a talk that was talking about transmission types and was talking about how they were seeing some very different transmission with rhinovirus compared to some of the other viruses they looked at, specifically coronavirus. And so it's interesting to note here that rhinovirus is still around in the population, he says. I've been wanting to look into rhinovirus rates more, and Andy helpfully has given me the data. So I think that's something I want to look into, and it stands out to me that I just heard this yesterday and was sort of, that's curious. And now here it is again with these data.
Well, you wouldn't expect them to go away entirely, right? They're still there. They're lurking somewhere. Enterovirus D68, we were supposed to have an outbreak in the fall of 2020. It's every 2 years. The last one was 2018. We did not have one. So it's probably a consequence of all the non-pharmaceutical interventions, I guess. But it's interesting that what he says about the going to a primary care, that would change it. But of course, if you use the hospitalization data, that's really going to tell you what's going on. That's cool.
So the message I'm getting here is that, yes, indeed, as we discussed previously, the social distancing, mask wearing measures, all of the anti-pandemic measures that we're taking may impact other infectious diseases, but it's complicated because our whole approach to healthcare is different as well. Yep. So the normal methods that we use to count these things don't necessarily apply in the same way. Am I reading that correctly?
Yeah, I think that's right. What it means is that society as we know it needs to fundamentally change, and then we will have far fewer respiratory infections. No more restaurants, bars, sports events. Stay home. Everybody needs to just stay home.
Everybody stay home, sit in their basement and podcast. Is that right?
Well, you don't have to podcast, but obviously that's unrealistic. But we already knew that the infectious diseases are a reflection of, because when we were hunter-gatherers, we probably didn't get many infectious diseases, at least from other people, because we never saw them. We're just
parasitic, not
I mean, we'd get them from eating. We'd eat the wild animals that we caught, and we get parasitic disease, and probably occasionally you get their viruses. But it's the way it is. It's fine. Rich, can you take the next one, please?
Robert writes, Dear TWiV, in response to the letter from listener John Jaros on episode 709 regarding concerns about lack of SARS-CoV-2 vaccine availability in low-income countries, I wanted to highlight the work of the COVAX initiative led by CEPI, Gavi, and the WHO, and gives a link that is COVAX Explained, and it's I forget what Gavi stands for. We'll look that up. In short, this program aims to solicit funding from higher income countries to support equitable distribution of vaccines in low income countries. They are currently aiming to distribute as many as 2 billion doses by the end of this year. I took it as a very positive sign that President Biden's Secretary of State designate, Tony Blinken, announced during his confirmation hearing that the U.S. would be joining COVAX, something that was, needless to say, not aligned with the policies of the previous administration. Yes, the previous administration withdrew both from the WHO and from COVAX. And one of the first things that Biden did, Gavi is the Global Alliance for Vaccines and Immunizations. It's a subgroup in the WHO. I also wanted, as a proud U.S.-Canadian dual citizen, to mention an initiative from Canada to donate surplus vaccines to low-income countries. Canada placed advanced orders for over 400 million doses of vaccines. Holy cow. But as a population of under 40 million people. While this may seem greedy, these orders were placed well before efficacy data was available, meaning these just as easily could have turned out to be bad bets. Assuming several other vaccines still in development turn out to work well, I suspect some other high-income countries may also have a surplus that could be in and could be in a position to donate surplus doses. Lastly, I'll mention that while these efforts by COVAX, Canada, and other generous countries are certainly admirable, they are only expected to provide coverage for 20% of the populations of low-income countries, mainly frontline healthcare workers and others at very high risk. It is certainly true that we have a long way to go to ensuring vaccine access to every human on the planet. Keep up the great work. Robert, who's writing from San Bruno, California, on a clear evening, 48 degrees Fahrenheit. Yes, these efforts to reach the less able countries are admirable, and I hope that they achieve some success. It is difficult, but I'm glad it's happening.
I don't know if you order 400 million doses if you're going to get them anyway, right?
Yeah, exactly.
You might know.
Maybe, yeah.
All right, Brianne, can you take the next one?
Sure. Chris writes, "Hi all. My parents, both in their late 50s, recently contracted COVID. They are still in the first week of infection and have been approved to receive Bam Bam, not even going to try and spell the full word. My issue is that my dad is very hesitant to receive the treatment because he read on the FDA fact sheet that monoclonal therapy could reduce the body's ability to fight off a future COVID infection. Could you explain this hypothetical risk further? I'm really concerned about him and think that the risks of not receiving the treatment far outweigh the risks of receiving it. Thank you all so much for your work on the show. It has been a real lifeline throughout COVID. Sincerely, Chris from Austin, Texas." He can hang out with Rich.
What?
So I think the idea here is that the Bam Bam antibody, as Chris mentioned, is a monoclonal antibody that binds to part of spike on SARS-CoV-2 and helps to neutralize it. This is a type of passive antibody therapy. You can tell that I taught passive antibody therapy in the past week in my immunology class. That basically is providing antibodies to, in this case, Chris's parents or whatever patient to help them fight the infection. But it does not involve the patient's body making their own B-cell response and making their own antibodies. In theory, these antibodies will clear the virus before the patient's own immune response will happen. And so if these people get infected again, they would be making a new primary response. It would be as if their body had never seen the infection. They would not have any immune memory.
Yep.
Right. So the Bam Bam is a stopgap measure. Okay.
Exactly.
And my God, you go for whatever they're offering you.
Take it.
The last thing you want to do is actually get the disease. So at least you can, with the monoclonal, you can put it off for 3 months. And then by then, 3 months from now, the vaccine's probably going to be a lot easier to get. All right. But don't mess with the disease. My God.
Yeah.
Right. And
exactly.
and Daniel has said, I think even that they're not being so compulsive about people having had monoclonal therapy, or maybe I'm thinking about people having had the disease, but has, never missed a chance to vaccinate. And so that if it came up even before the 3 months, he might say, go ahead. But just to, again, clarify what the FDA fact sheet says, that monoclonal therapy could reduce the body's ability to fight off a future COVID infection. That would be relative to if the patient got no monoclonal therapy and produced their own immune response to the natural infection, but with whatever other consequences might come along with that. And so I think that's the only way they can say it's going to reduce your ability. They have to mean relative to if you didn't get it and your immune system created a response to the natural infection.
Yeah, it's not reducing your body's ability to fight off a future COVID infection compared to a person who had never been infected before.
Right.
You're still in that same naive category. You just don't get that extra memory bonus.
Right.
But you should take it because it could save your life.
Absolutely.
Yes.
All right. Marvin writes, Dear TWiVers, I see many SARS-CoV-2 variants containing geographic references, UK, South Africa, Brazil, Ohio, etc. Why? References to China, Wuhan, and the like were long ago and prominently replaced by SARS-CoV-2 and or COVID-19 as applicable in TWiV Microbe TV and elsewhere. I love that. TWiV Microbe TV and the rest of the world. Marv is in 4 degrees in rainy Vancouver. You got me. That's what the news likes to do. And all I can say is I'm glad they're calling them variants. Thank you for that.
Yeah.
Not strange.
Even I like to do it because the names they give them are impossible.
Yeah. B117, something else, 135 point something. Yeah.
Yeah.
Oh, the EEC one, the amino acid change that's E488K, which goes by the nickname of EEC now or something. But yeah, numbers are hard to remember. And I think that's probably why.
I mean, we prefer not to identify them with places, and that's been long-standing in virology. You know, see Nombre virus, right? They tried to name it Muerto Canyon virus, and the local people, they didn't want to do that. So totally understandable. But you know, I'm glad we do the right thing here on TWiV 1B1117, or you know. Sure.
Yeah, part of being able to remember it by geography is that I picture a map in my head. Okay, and it helps me. It just helps me keep them sorted out. Whether it's kosher or not is another question.
Kathy, you're next.
Isabella writes, Hello, I just recently began to follow your podcasts, and I have really enjoyed the variety of topics you and your team cover, especially as it relates to SARS-CoV-2. I'm a postdoctoral researcher at Colorado State University, and I love listening to the podcast on my long drive home into the mountains. My favorite podcast thus far has been episode 708 for 2 main reasons. One, the discussion about nanobody treatment and camelid antibodies was so intriguing. Before becoming a veterinary researcher, I wanted to specialize in camelid medicine as a practicing veterinarian. I think alpacas and llamas are the most interesting critters on the planet, and it's great to see all the attention they are getting regarding their unique anatomy and immune system. A little tip I would like to share, an easy way to distinguish alpacas from llamas, is to look at the shape of their ears. 2, I really enjoyed the conversation on inactivated vaccines against SARS-CoV-2. It was great that you and your team discussed some of the history regarding the use of inactivated vaccines. I do hope you all revisit this topic as more data comes out for the use of inactivated vaccines to fight this pandemic. You are correct that formalin and beta-propiolactone have been evaluated as methods to create inactivated vaccines. Our lab is currently looking into a new method using UV light and riboflavin to inactivate pathogens for vaccine production. UV light and riboflavin have been used extensively in the blood transfusion world to reduce the risk of pathogen transmission by blood transfusion. Now this method is being tested as a novel way to create inactivated vaccines. We hope our work will demonstrate the value of inactivated vaccines in this fight against pandemic viruses. I hope you and your team will revisit the topic of inactivated vaccines soon. A great yet very important topic. Thank you for all the work you put into this podcast. Warm regards, Isabella, as we said, a postdoctoral researcher at Colorado State University and a veterinarian, DVM PhD.
Hey, Isabella, if you liked that episode where we talked about llamas and alpacas, you have to go back and listen to the episode with Laura Splann, episode 691, who is a science artist who also spent a lot of time thinking about camelid antibodies and also got some wool from alpacas. And it was all very fascinating.
Oh, that was so cool. Wasn't that great?
Mm-hmm.
Yeah.
That was so great.
So Kathy and I both went to correct a grammar thing at the same time. We're the two that see the spell check, underline something. We got to fix it. Rich, you're next.
Andrea writes, Hello, TWiV team. I already expressed my appreciation for all you do in your last email. Thanks again and keep it up. As I mentioned in my last email, I work for an agency serving people with intellectual disabilities. Thankfully, all staff and individuals are included in New York's 1A tier, and most have been vaccinated with the first shot by now. I discussed the vaccination with a colleague yesterday, explaining the difference between sterilizing immunity and prevention of disease. In my own layman's terms, I described that most likely the virus will have a little time to replicate in the oral cavity before it's being recognized by antibodies in the bloodstream. Hence, people may still spread the disease while not getting sick. He brought up an interesting question. Does that mean that people who become infected after a vaccination can still lose their sense of smell and taste? If so, knowing that up to 80% of people who develop symptoms also have a loss of smell and taste, could this be used as a marker to pick out individuals with a latent infection? I'd be curious as your thoughts on this. Thanks, Andrea, who is an RN and what is a BS in?
Bachelor of Science in Nursing.
Okay. Well, ain't that interesting? I don't know. I guess we'll find out.
I mean, if I remember that one of the first things many people have is a loss of smell or taste, and then they go and get a PCR test and they're positive, and then they go on to get other symptoms and signs. But I don't know if that would happen in a vaccinated person or not. Yeah, I don't know. We don't know.
But I think it's an interesting thought.
Doesn't it seem like from some of these vaccine trials we would maybe have some data on that?
Yes, we haven't seen it, have we? Yeah.
Have they been looking for loss of smell and taste in the vaccine trials?
I think it's probably one of the signs, the potential signs that would trigger a PCR test, right?
I would think so.
It would make sense.
We'd have to really dig into the various trials.
Let me look in the paper for today. Meanwhile, address this bloodstream antibody. That's not right. Okay.
Yeah. So, well, first of all, we'd probably be seeing virus in the sort of nose as opposed to the oral cavity, but that's minor pedantry. Yeah. So it's probably not so much about antibodies in the bloodstream as antibodies that are actually in some of those mucosal surfaces.
Yeah. So the Sputnik paper has no mention of loss of smell or taste.
I know I skimmed the Moderna and Pfizer data recently, but I was not looking for loss of smell or taste, so I'm not sure. I could have missed them because I wasn't looking for them.
That's a good question. I don't know. All right, one more, Brianne.
Sure. Daniel writes, "Hello, TWiV. Writing from Arlington, Virginia, where it is 39 degrees and we keep missing out on snowstorms. You can have mine, Daniel. Have been a regular listener since hearing about your podcast on Slate Political Gabfest last summer. A couple questions. One, I know you touched on this a bit earlier, but can you please explain why the vaccine will still work on all the mutations out there, assuming that this is true? To that point, do you have a view on when the worst case scenario endpoint is, after which some version of normal life will return? My understanding is that even assuming a relatively slow vaccine rollout between vaccines and natural herd immunity, we should get there by late 2021. Is there some nightmare scenario where the mutations essentially outrun our ability to vaccinate and we are stuck in the pandemic indefinitely? And number two, can you comment on the risk that mutations will make the virus more deadly for children? I haven't seen anything reported like this, and my, albeit quite rudimentary understanding is that the structure of the virus makes it more difficult to infect children, so presumably it would take quite a lot of mutations for that to change. Or is that just unknowable? Thanks again for all the work you do. We rely on you guys for a real understanding of these issues during the pandemic. Dan. So as far as point one, Dan, so you ask about whether the vaccines will work on all the mutations out there. we probably don't know about all the mutations out there. They have not all been described. But what we're able to do right now is look at some of those mutations that have been described and see where they change amino acids in the virus, if they change amino acids in the virus, and how that corresponds to places where antibodies or T cells, mostly antibodies, recognize the virus. And so what we can see is that the main places where antibodies are contacting the virus, the epitopes, are not changed compared to other parts that are changed, but that's not where antibodies bind. We can then also do experiments where we take antibodies from people and actually test them against the variant viruses to actually see whether we think the vaccines will still work. And that makes us think that the vaccines should still work rather well against most of the variants that are out there. There are some that reduce immunity a little, but it's not clear that that's enough to actually matter. But I do not think that I can comment on all of the mutations out there, because I know that there are many that have not been described that I can't comment about.
The B1135, and I will not mention the country, is the most concerning to some people, because it does reduce. It is not as well neutralized by sera from vaccinated people. And I think AstraZeneca just stopped distributing its vaccine there in that country as a consequence. So that's kind of concerning, I think. And I think that we need to get everybody vaccinated as quickly as possible, right? Yeah. That would help cut down transmission.
Yeah, Dan asks about some nightmare scenario where mutations outrun our ability to vaccinate. There's a lot to unpack there, but what I will say is that one of the ways that the virus gets more chances to mutate is that it reproduces more. And so as we have more infections, the virus gets more chances to mutate. So the more we can use our non-pharmaceutical interventions to stop the virus from reproducing, the more we also stop mutations and stop that nightmare scenario from happening.
I just want to reemphasize something we've said before, and that is that when you mount an immune response to the virus, you don't make just one antibody. You make a bunch of antibodies with different specificities. That is, they'll glom onto different parts of the spike protein and compromise its function in different ways. Not only that, but you make T cells that bind to different epitopes as well, not only on spike, well, in a vaccine, just on spike. So if you change just one part of the protein, it doesn't necessarily change the ability of all those other antibodies to react with it. It may change some critical antibody sufficiently so that the effectiveness of a vaccine might be reduced, but the chances that one mutation is going to completely neutralize the effectiveness of a vaccine, in my mind, is pretty slim, okay? And what we really care about is whether it's going to keep you out of the hospital. And in the meantime, as Brianne said, we need to stay on top of this to minimize the chances for these mutations arising. But it's clear that it's clear to me now from several things that we've done, including the B113 mutation arising. Is that what it is? 113?
135. 135.
135. See, I can't do that. I can visualize the country. It's clear that there can be antigenic variation in this and other coronaviruses. And so we're going to have to be vigilant.
I have to say that if you wear masks and do physical distancing and all that stuff, that will reduce your chance of being infected. So the problem now is that not everybody is doing this, and we've got a lot of hosts to infect and mutate and recombine in. So, we could deal with it a little better than we're doing. What do we think about this second part?
Well,
I think we don't know.
I don't know any evidence that this is becoming more deadly for children. Do you? No, he
No.
said, I haven't seen anything reported like this. I have not. And I don't know why. Why would it make the structure make it more difficult to infect children, right?
Right. I don't know any evidence that it's more difficult for it to infect children. And I also, this we've talked about before, it's hard to imagine selection for something that makes it, more deadly is a pretty broad term. So I suppose I could cook up some way that you could change the virus in a fashion that was advantageous for the virus that could make it more deadly for children. But it's not something that concerns me.
No, I haven't seen a human virus become more deadly over time, and I've been looking at them for a while, so I don't think that's going to happen. Although you can never rule out, right? You'll always be surprised, but I don't think it's likely. I'm more concerned about the antigenic variation. Okay, time for some picks. Brianne, what's up this week?
So I have an article that I read that just made me really happy. And it may be because of the fact that I've been spending a lot of time, worrying about classes and getting ready for teaching and feeling guilty when I do things that are outside of class prep. But this is an article called "Do Scientists Read Enough Fiction?" And this is in EMBO, but it's an open access paper where someone writes about all the ways that he thinks that reading literature and fiction has been important for his academic life and helped him as a researcher in terms of both helping him with his writing, helping him read. As I thought about it, and I thought about lots of ways that it helped me with sort of being able to be creative and thinking about different ways to think about science. And I think that, and he gives us a bunch of citations about how reading novels improves brain function on a variety of different levels and things like that. And I think that at the end of this, I felt much more compelled to block out some time every day for reading some fiction. Because he reminded me of all of the benefits. So I hope that everybody else can get that too.
Does he have a favorite genre?
Yeah.
I haven't read this.
He cites a lot of different books that he likes. And so most of the things he talks about are classics. He says that he sometimes tries to revisit War and Peace, for example, but he mentions a lot of different fiction here.
Okay.
I haven't read fiction in over a year because I used to read fiction when I was traveling, right, on airplanes and stuff, and I don't do that anymore, so don't read it. I don't even remember what the last fiction book I read is. I was just looking at the Kindle and trying to I have no idea. Anybody read? I know Alan reads fiction because he makes picks of the week a lot about that. Anybody read any fiction lately?
I last read fiction over sort of the holiday break. I was reading some science fiction novels, Ready Player One and Ready Player Two, but I've had another one queued up, ready to go, and then the semester started.
Oh, I know.
But I'm going to make time for it now.
The most recent fiction I read was that Nicholson Baker article on the origins of SARS-CoV-2. There you go. That was fiction. And if
So
you're
what
mad,
was that, Brianne? Ready Maker 1 and Ready Maker 2?
Ready Player One and Ready Player Two.
Yeah. What sort of sci-fi is that?
They are books about people playing an immersive video game where they sort of can be immersed completely in this video game.
didn't you pick
so
that
have you
recently?
read Ender's Game?
I love Ender's Game.
Okay.
And yes, I picked Ready Player Two recently.
Yeah, I thought so. Sound familiar.
Where the characters have to think a lot about the empathy of being others in a video game.
You know, it's funny in that article they say if you caught a PhD in the middle of the day reading fiction, you'd say wasting their time. I never would read in the middle of the day in my office fiction. I would feel the same. I would feel so guilty. Absolutely.
Yeah, he talks about that here, how now he sort of blocks off some time every day, sometimes in the middle of the day.
Hey, my colleagues here,
writing.
Saul Silverstein used to take a nap in the afternoon, right? And I said, what the hell? He said, you need a nap. It refreshes you. And that's like totally counterintuitive to how I've been raised. You don't take a nap in your office in the middle of the afternoon. But, I'm screwed up. What can I say? Kathy, have you read fiction lately?
Yeah, I've read a couple of things by Rhys Bowen. It's a series I like. And also Where the Crawdads Sing. I can, because I say read, and it's really in quotation marks because I listen, but when I go out walking, if I'm out of podcasts, then I can get back to my fiction. And I can remember like where I am at certain times when certain plot things happen. So I remember I was doing some stuff in my garden at a really key point in Where the Crawdads Sing. And so I'll never forget that. Cool. But then I get into some longer other things, like I'm listening to Barack Obama read his most recent book. And that's many, many hours of listening. So, yeah.
Rich, what do you have for us?
I got a couple of slow-mo videos of insects flying. So these are two videos from the same guy. I'm looking at this. His name is Adrian Smith, and he's the head of the North Carolina Natural History Museum's Evolutionary Biology and Behavioral Research Lab. And I was just going to do one of these, but they are more or less non-overlapping. He's got this little lab he set up, I think he says, in his laundry room. Okay, the little home lab to take slow-mo videos of insects that he picks up from his yard, or puts out a light at night and collects a bunch of insects under a tent. And he's obviously an entomologist or something like that, because he knows what all these critters are. And they're just great videos. You can watch these guys take off and fly around. A lot of it, and his narration is pretty good too, because he's pretty humorous. He is the nerd's nerd, I must say. He's really great. But a lot of these are pretty ungainly looking flying machines. And I never realized how beetles' wings fold up under whatever he gives the name for it. The external wing is called. So anyway, just fun videos.
Cool. Yeah, Rich, do you kind of browse YouTube, or do you get these things suggested to you?
I don't know where this came from. No, I don't browse YouTube usually, though. Where I wind up in YouTube mostly for a cappella singing or other things, and I can fall down that hole and probably wind up with insects. I forget where this came from. It may have been a Facebook thing or something like that.
I usually go on YouTube to find out solutions to tech issues I have, recording or whatever. And it's funny, I'll be watching a review of a piece of equipment that I need to master, and then they give you recommended videos, and they give me TWiV. It's like, where does that come from? I'm happy to see that they're recommending that to people who are watching tech videos. That's cool. Kathy, what do you have for us?
I picked something that Justin sent to me. So thanks to Justin. It's called Badger Seal, which sounds like a mashup of two animals, but comes from the University of Wisconsin, and they are the badgers, of course. And this is a device that helps you seal your mask better. It's also called a mask fitter. And so it is kind of a wire thing that you can change the shape of it. And it's got two different designs, and you can purchase them. If you're part of the University of Wisconsin-Madison, you can just do it on a normal university request form and list badger seal and get them. But basically, a part of it can either, the adjustable part can be behind your head or the adjustable part can be below your chin, but it makes the mask fit better. And so there's original and ear loop. And this is important because of the increased need for really good masks. And it also supposedly can help with fogging. So that could be important for some people still. So there's a couple of videos, and it says you can, there's a do-it-yourself, and they have open source design with detailed steps. I haven't looked at that. But anyway, it seemed like a kind of a additional mask-like thing. So
Cool.
check out.
It's this mannequin that exhales fog. Look at that, how it leaks out of the top. My gosh.
Yeah, it's dramatic, showing you how,
Wow.
But when you put the badger seal on, then it's better. So
Interesting name, badger seal.
mm-hmm.
All right, I have well, if you have seen lately, there's been two postal workers on the news with a TWiV T-shirt, and they even had a TWiV mug in the back, and people have been saying, where do I get these? So you go to the Microbe TV store, cafepress.com/TWiV, and we got masks, we got T-shirts and hoodies and zip hoodies and bags and mouse pads and mugs and stickers and buttons. We even have a button that says, mice lie, monkeys exaggerate, and ferrets are not people. And that was made by Christian years ago. And Christian recently made a image of Daniel Griffin, where it's a silhouette and it's really nice. And it says Daniel Griffin, licensed to heal, right? It's kind of the knockoff of the James Bond. And Cafe Press won't allow me to put it on merchandise. They say it rips off 007. So I'm trying to argue with them about that, because a lot of people want that on a sticker. Anyway, that's a good way to advertise the podcast.
You could actually outfit every part of your life with TWiV GAC here.
You could.
Yes.
I add stuff. Yeah.
Wait,
I
you don't have
vouch for the
socks?
hoodies.
You like the hoodies, right? Yeah.
I vouch for the hoodies. Yes.
Yeah.
You need
people buy
socks.
Socks? I don't think they have socks. No.
Yeah, well, they may not, but then,
I could expand to there are other places that do this. This is one I did in 2007. Wow, way back when. And I just left it there. And there are other places that have good stuff too. I should probably broaden them and see if they'll accept the Daniel Griffin, because people want Daniel Griffin stickers. All right. That's TWiV 718, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. If you want to support us, well, you could buy some swag. We get like $1 per swag item. Or you could, but those are mainly to advertise, right? We get on the news. It was like on NPR, these two people wearing it's cool. I got a lot of people emailing about that. Microbe.tv/contribute. Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
thanks. This is a lot of fun.
Rich Condit's an emeritus professor, University of Florida, Gainesville. He is currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Brianne Barker's at Drew University, BioProf Barker on the Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology. I'm going to say WS. Blog stopped working. Virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a couple of days. Another TWiV is viral.